The synapsid reptile lineage, which would include the ancestors of mammals, split from the lineages of other reptiles by the mid-Carboniferous period. Over almost 100 million years, reptiles evolved into mammals and evolved a number of changes during this time period. The first mammals evolved by the late Triassic period, but it wouldn't be until the extinction of so much of Mesozoic life, including the dinosaurs, at the end of the Cretaceous, when, quote, the age of mammals, the Cenozoic era, would begin when placental mammals diversify to such a great degree, and that would be another 150 million years after the first mammals. So this is an incredible span of time. During this time, mammals, and then particularly the live-bearing mammals and the placental mammals, they evolved a number of changes to their brain, particularly the cerebrum. First, overall, the brain became larger, and part of this was because mammals evolved a number of adaptations to get more energy. They improved their breathing, they improved their ability to chew food. This not only would power their warm-blooded metabolism, but also their larger brains, given that brains are energetically very expensive. Secondly, the cerebrum is a region of the brain contained in all vertebrates, but it became a much larger part of the brain relatively compared to the size of the cerebrum in other vertebrates. Mammals are not the only vertebrates to possess a cerebrum or telencephalon, as is seen here. The aqua region of the brain is the telencephalon or cerebrum. So clearly this is present in fish and amphibians. But the cerebrum is composed of separate regions, and one region known as the dorsal pallium, pallium refers to the roof, what makes the roof over the cerebrum. This is also known as the neocortex. That's depicted here in pink. So notice, while this pink neocortex or dorsal pallium composed part of the cerebrum of a frog or a fish, it was not the dominant part. Whereas when you look at mammals, then this pink dorsal pallium neocortex region becomes more and more dominant as you go from basic mammals to placental mammals and then into primates. So the human brain is composed primarily in terms of area of neocortex, not just the cerebrum, but a specific region of the cerebrum. Finally, and this is incredibly important, the neocortex, the dorsal pallium, this pink region in a mammal is not that equivalent to those of the non-mammals. It is fundamentally different in its cellular cytoarchitecture. In other words, if you look at it under a microscope, the distinguishing feature is that there are 6 layers of cells in the mammal neocortex, whereas in typical cerebral cortex tissue, there are only 3. That means that there is an incredible increase in the number of cells, but also in the complexity of the synapses. So this change at the microscopic level, where the neocortex, the pink region, the dorsal pallium of a mammal, is so much more complex than that of, say, a reptile, or even of other regions of the mammalian cerebrum that do not have these 6 layers. This then gives the cerebrums of the mammals an incredible increase in complexity compared to other organisms.